All right, ladies, just wait 1 second. We'll wait a few minutes until we find out, how many more people are coming. So we'll wait a few minutes to see how many people show up, okay? So today we're going to talk about, finish a little bit about just certain concepts in 2. The rest of chapter 2 is going to be a lot of Gen Chem. So watch the videos I have. It's pretty detailed. And then I want to get into chapter 3 material in the second hour or so. So just a little warning that we will go about an hour and a half or so just talking about this material. And keep in mind, I did post the assignment for chapter, actually, let me see if I posted it, if it's up now. Let me see. Assignments. Okay, that's on the wrong one. Right, so the assignment for chapter 3 will be up this afternoon, right? So we will have a connect assignment on chapter 3. Chapter 3 is a little bit more difficult. So, with chapter 3, there's a lot of material, especially towards the tail end, about genetics, about DNA replication, about protein synthesis. Perfect. Tell me if you can hear me. I know Megan's having a little issue here. That's okay. Trying to figure out some of the content here. And in a second, once everybody's here, we'll get started. All right. Or once some people are here. Okay, let me take a look. No, I don't have much yet right now. I'm waiting for people to see if anybody shows up, and then we'll get started. That's all. All right, so I'll just wait another, well, actually, we'll get started in a second right now. So with chapter 2 material, it's already 10 minutes up, so I want to make sure we get into chapter 2, right, and chapter 3. Now, just give me 1 second until I have that screen up, and it should be up right now with chapter 2. Let me just share the screen. Okay, so chapter 2 material right here. Go full screen here. Now, when we're looking at the end of chapter 2 material, the beginning we already talked about, right? Again, all the videos I have already online should be actually, we kind of discuss it a little bit more detail in the actual classes than what we will have today. So just a little warning, people want me to post the videos of us kind of voicing over and having our class lecture like this. The problem is we don't have as much time as in a normal semester when we'd have 3 hours. So I can go in a little bit more detail with those videos and go in depth in those videos. Now, towards the end of our class, we talked about bonding. Now, this should be a review of your Gen Chem, right? This is not something that should be really difficult to discuss, but if you have not had Gen Chem in a while, you might need to read up on it because we only need to know the basics. Basically, what is the difference between a covalent bond and an ionic bond? And more importantly, how about in covalent bonds, what kind of covalent bonds do we have? We have nonpolar and polar. Covalent bonds should be sharing of electrons between 2 different atoms or elements. And when you have sharing of electrons between elements or atoms of equal strength or very, very equal, almost equal strength. We're going to have a nonpolar covalent bond. Now, how can we tell if we can have equal strength or not? We're going to look at a chart like, let me see this chart right here. This chart is for electronegativity. And what we're going to see is the darker the red, the more ionic bonding it will go through. So you see something like fluorine, right? Fluorine has an electronegativity of 4. When it binds with a hydrogen with 2, what's going to happen? Well, this one's almost twice as strong. It's going to steal the electrons from hydrogen instead of sharing it. That will make it a ionic bond when this and this reacts. Now, how about oxygen? Look at how strong oxygen is. It's the second strongest. The thing about oxygen is it can't really steal 2 electrons. So what it can do is actually bond with something like hydrogen with 2 covalent bonds, but because it's so much stronger, it's going to pull both of the electrons from hydrogen. So then there's an unequal sharing of electrons. That's why with H2O, you'll always see a little bit of an electron pull towards hydrogen, towards oxygen. The oxygen becomes slightly negative with the weird delta sign on it. And then hydrogen's getting some of its electrons removed. And what happens then? Well, it starts to lose some of the electrons, so it is partially positive then. And I think that's where we ended. So here's a picture of water. Oxygen in red, hydrogen in blue, H2O. What happens? Because of the way the electrons are being pulled away from hydrogens on each side, the oxygen right here has a partial minus charge. This sign is Greek for delta, and it's supposed to represent a partial charge, not a full charge. The only time you see a full charge is if the oxygen actually removes the electron physically from hydrogen. That's not what happens. Here, it's just pulling the shared electrons towards itself. And what happens? The oxygen right here kind of hogs the electrons for itself and doesn't share it equally. Because it's hogging it for itself, it's like it's gaining a partial charge. And that's what we see. You're gaining an electron, you're gaining a negative charge, but it's not a full negative charge, it's only a partial. That's enough though to start interactions between these different hydrogen molecules from different water. So here's one H2O molecule. It's going to start the partial positive. Oops. Right. The partial positive from that water. Where is it? Lost it. The partial positive from the hydrogen and water is attracted to the partial negative from the oxygen. So these two have this magnetic electrical pull from one molecule of H2O to another. This is a hydrogen bond. By itself, it's not very strong independently. Compared to an ionic bond, compared to a covalent bond, they're not very strong. Now, when you see like in a droplet of water, there's trillions of H2O molecules. You're gonna have trillions of different hydrogen bonds between them. The additive strength is what makes hydrogen bonds so important. The numbers game, right? The more of it, the more strong the bond is. Now, let's talk about dissociation. And I think we talked a little bit about dissolubility before we left. When you put a salt in water, you can completely dissolve it. And that's what happens here. The sodium chloride in the salt, right? Sodium has positive charge. Water has oxygen with a partial negative charge. So the sodium is surrounded by the oxygen in water. We say it's solvating that sodium. The chlorine as well. Chlorine's negatively charged. Remember, salt is sodium chloride, right? When they react, they form the full charges. Chlorine receives the hydrogen from sodium. Sodium lost the electron. As it lost the electron, it lost a negative charge and becomes positively charged. And what happens then? Sodium gets surrounded by the negatively charged oxygen in water. Chlorine gets surrounded by the hydrogens in water. Why? Because opposites attract, right? You have a negatively charged chlorine, and now you have all these hydrogen molecules with its partial positive charge. Now they're opposites. And what we're going to see is the water molecules right here will then separate the sodium chloride, and it completely dissolves. And once we dissolve it, then you have the sodium and the chlorine separated. That's an electrolyte. Remember, the only difference between a salt and an electrolyte is that the salt is what we call it when it's like a cube. And then once it's placed in water and it dissolves, it's an electrolyte. Okay, now in terms of chemical reactions, understand the difference between synthesis reactions. Synthesis builds anabolic steroids, anabolism. You're usually going to make something larger from something smaller. In order to do it, we also call this reaction a dehydration reaction. When you have a dehydration reaction, water is removed, combined from both starting material. So here you have two amino acids, and now we're going to remove H2O. As we remove H2O, the OH from this first amino acid and the hydrogen from the second amino acid is removed. In the process, you can see it's even color-coded, right? You have the H2O being removed, and now by removing the oxygen here and the hydrogen, this carbon has an extra bond that it can handle nitrogen as well. By removing this hydrogen, this nitrogen has an extra bond it can link to. And now we're going to link these two together. This is a peptide bond. And this is how we start making amino acid chains that will turn into proteins. So when you look at anabolic reactions always build. Now, don't think about anabolic and anabolic steroids and it's only building muscle, right? It's going to be there for amino acid and muscle production, as well as production of carbohydrates, right? Glycogen. For plants, you're going to have starches. All of them start with just simple sugar, and then we add more and more sugars to it. On the opposite end, catabolism is a breakdown. And if synthesis reactions and buildup reactions require you to remove hydrogen, breakdown reactions put water into the solution. So here's a disaccharide, 2 sugars. In order to separate it, water is used and an enzyme is used. In the process, you break apart the 2 sugars into 2 glucose molecules. This is maltose, and with water and maltase, the enzyme, then we're going to destroy and break apart this maltose disaccharide into its 2 glucose molecules. Keep in mind, when we're eating starches, like in a potato, we're eating a lot of these glucose molecules bonded together. Now, it's not just 2 of them. You're going to have thousands of them. And then there'll be branch points with other thousands of individual glucose molecules bonded like this. And then you should see another 2, and another 2, and then another 1,000 glucose molecules. That's what a starch is. Now, why am I mentioning this? The only way we can absorb the glucose is if we break it down from a few 100,000 molecules of glucose bound together. In order to absorb it and bring it into our cardiovascular system, we need to just have individual glucose molecules. Even if we break it down and we have 2 of them, and this is our leftover, we cannot absorb this disaccharide. We need this last step to separate even this small disaccharide. As we separate it, now we have 2 individual glucose molecules. Perfect. Now we can absorb each one. But when they're together, you can't absorb any. So our body's proteins are that specific. All right. Now, let me go through so you guys can watch that about the electrons transfers right there. Now, when we're looking at mixture and medium, right? Basically, I'm skipping around because I don't think we have as much time to discuss it here in our class. So I do have the video on it, right? I want to talk about a mixture, mixing medium. What is a mixture? What is a suspension? What is a colloid? What is a solvent? What is a solute? And then we're going to talk about osmolarity, right? Keep in mind in terms of mixtures, mixtures are substances physically, but not chemically combined. In other words, you're not breaking any bonds. You're just putting something in water, in some other kind of solvent, some other kind of liquid besides water, right? Water is the universal solvent. Usually for everything, we use water. But there are times in organic chemistry where the solvent, the liquid part, is not water, right? But that's pretty rare. Now, in us, we're going to see suspension in our body. We're going to see colloids in our body as well. What is a suspension? What is a colloid? Sorry about that. A suspension is a mixture of materials where the materials separate unless it's stirred. Right? They talk about sand and water, right? Even better yet, have you guys ever seen freshly squeezed orange juice? What happens in freshly squeezed orange juice? All the pulp is going to rise to the top, especially if they crush the pulp already. And in order to mix everything around, you have to shake it. That's a suspension. Now, an example of this I always use in our class is antibiotics, especially for babies and young kids. You can't give them a pill to swallow. So what do? We give them a liquid that has the actual medicine in it. So you fill up your prescription. Let's say it's for Augmentin. And when you go see the pharmacist at that point, when you come in and get it, all you see is just a bottle. But in reality, it's just dry Augmentin, right? They put some additive in there to hopefully make it taste a little better. And when you go in, they add water to it, distilled water. As they add distilled water, they shake it up. So then all the drug is found throughout the canister, right? Now, what happens if you go home, wait a couple hours, and then give your son or daughter some of the medication? Well, if you don't shake it up again, when you try to give it to them, depending on where you put your syringe to draw out the solution, right? If you draw out the solution of the antibiotic from the top and you didn't shake it up, you're going to have less drug than you imagine. The reason for it is this: the actual Augmentin is heavier than the water you put in there, right? Then the drug, because you haven't shaken it in the last few hours, the drug starts to fall towards the bottom. If you don't shake it again before you give the drug to your son or daughter, if you take that medicine from the top, well, all the drug fell down to the bottom. So you're not getting as much of the medicine as you think. So every time you're about to give somebody some antibiotics, you need to be able to shake it a little bit and then start to, right, give it out to your child. Now, unlike the antibiotics, where you have to keep shaking it before you give it to them, there are other kind of mixtures called colloids. Colloids is like milk. Now, you will see people that shake milk. You don't need to shake milk. Milk has tiny fat particles that scattered throughout, and fat's lighter than even the water that's found in milk, the liquid part. And because of it, it's always dispersed throughout the whole area of the milk. You don't need to shake it. Now, solution, what is a solution? A solution is a mixture of liquids, gases, or solids. Now, in our body, we're going to usually talk about these, liquids and gases, right? In 202, we're going to talk a lot about the gases, especially in our environment. In 201 and 202, we talk about the liquids a lot. So blood is a mixture of liquids, where the fluid, the solvent, is plasma, made out of primarily water. So solvent in our body is usually water, and it is the fluid that dissolves the solute. The solute in our body is any solid. It can be a salt. It can be a protein. It can be cells. Any solid that dissolves when mixed with the solvent. Whole blood is a mixture, a solution, and a colloid. Now, when we have, let me go back here. When we have a solution, we need to know the amount of material in that solution, right? We need to know how much solute do we have. Why is that important? Simple. What happens when you go into the ER and you are dehydrated? Does a nurse or the doctor just gives you a 2 liter of water and tell you to drink it? Hopefully not, right? Now, why don't they just give you water to drink, especially if you're thirsty and you're dehydrated? What we're gonna see is if you drink too much pure water, it can lead to water toxicity and even death if you don't let yourself urinate. So water is not something that is, it's usually a positive thing if you drink a lot, but it's only positive if you let yourself urinate. Now, most of us are not going to hold our urine for super long amount of time, but there was a time when there's a contest for Nintendo Wii where somebody held their urine for hours after drinking gallons of water. Eventually, she won the Nintendo Wii. This was obviously in like 2005 or something, right? She won the Nintendo Wii, but she lost her life as a result of the water toxicity. So the last thing we want to do is just drink pure water, especially when you're dehydrated and you're in the ER. Why? Is because of this concept, right? Osmolarity reflects a number of solute particles dissolved in 1 liter of water, right? So let's say you go into the ER because your your kids are dehydrated. The doctor and the nurse will put a line into you, and they will put a bag of saline into you. The bag of saline already has the concentration of the salt, usually sodium chloride, and it has a volume. So it is already telling us what? The number of solutes and the volume. It's telling us the osmolarity already. And the osmolarity of that fluid should be similar to the osmolarity of your blood. So then when I introduce it into your vein, this saline solution that has the same osmolarity as my whole blood, that saline will then be incorporated immediately. As it's incorporated, then we're able to not even worry about any water toxicity, right? So that's the reason why instead of giving you pure water, they give saline. It's much more easily incorporated. And then you decrease the risk of water toxicity. Now, in our body, we use milliosmoles of solutes, right? In our body, the average of all solutes, we're gonna see, in our body, we're gonna have whole blood. That's one area where we see water, a solution, right? And we find it as part of plasma. Then you're gonna see the trillions of cells in our body, 38 to 44 trillion cells. We can't even agree to the nearest trillion. Each of those individual cells have a little bit of water inside. But when you add it up and multiply it by 44 trillion cells, any small amount of water, when you have 44 trillion, becomes a lot. So we'll have water intracellularly that also has 300 milliosmoles of solutes there. Because in our cells, we have organelles, we have lots of proteins, we have sodium, we have potassium, we have calcium, we have chlorine, all are solutes. And when we add up all the solutes inside our cell, we're at 300 milliosmoles of solutes. So we're in equilibrium, right? And then there's one other area where we see water, and that's the area between the cells. We call the interstitial fluid. Have you guys ever gotten a scrape where you're not bleeding, but you see this yellow liquid still ooze out? Right? Now, people think it's plasma. It's not plasma. If it was plasma, you would be bleeding. But the cut or the scrape is not deep enough to cut through your epidermis. So what happens? Where's that yellow fluid coming from then? It's coming from the fluid found between cells. We call that fluid interstitial fluid. That interstitial fluid surrounds every cell in your body. It surrounds the blood vessels, carrying that whole blood inside of it. So the interstitial fluid also needs to be in equilibrium. And we're going to see that the interstitial fluid also have a solute concentration of 300 milliosmoles. We're in equilibrium in all three compartments in our body where we see fluid. Again, the compartments inside the blood vessel, plasma, outside in the surrounding interstitial fluid space, surrounding every cell in your body, surrounding even the blood vessels, and then inside each individual cell. If we are in equilibrium, there will be no net movement of water in any one direction, which is perfect, right? So in terms of acids and bases, very general, understand the concept of acid releases hydrogens, base accepts it, right? Understand as well that the hydrogen concentration is inversely, right, proportional to pH. So the higher the concentration of hydrogen, the lower the pH. Right, now we'll talk more about buffers next semester. What I want us to do now is just kind of review some of the carbohydrates and the macromolecules, right? So come back here. So there's three main types of what we call macromolecules. Oh, come on. Let me rephrase that. Four main types: carbohydrates, lipids, proteins, and nucleic acids. So carbohydrates are, we mentioned before, right? Now the weird thing is this: carbohydrates, lipids, and proteins. Look at all they have in common. All of them contains carbon, hydrogen, and oxygen. See that? Why is that important? Is because there's a lot of interconversion. Carbohydrates can be turned into lipids pretty easily. Carbohydrates can also be turned into some amino acids pretty easily as well. Lipids can be turned back into certain type of carbohydrates. So there's a lot of interconversion between these first three macromolecules. We kind of understand this as well. What happens if, and you will see people who are vegetarians, yet they still are obese. Why? Well, if you're a vegetarian, you're just eating, you know, broccoli and vegetables. How do you gain so much weight? It's because you eat a lot of carbs, a lot of starches. If you eat a lot of carbs, a lot of starches, you can only store so much of it as glycogen, right? Human starch is glycogen, is what we store inside our skeletal muscles. Before you have a big game or a big meet, right? If you're a swimmer, if you're a wrestler, before you have any of that, usually people and teams go out for a big pasta dinner 2 or 3 days before. Why? Pasta has lots of carbs. You break it down, the carbs, and carbohydrates just look like this. Let me see if I have a long chain of it right here. Right here. Here's a long chain of the carbohydrate for glycogen right here. Take a look what they all come from. Individual glucose molecules. This hexagon is an individual molecule of glucose. Here's another one. Here's another one. We're going to have thousands of this going all the way back here. Then you're going to have branch points, and you're going to have thousands going up here. Then you might have another branch point down here, and then you have thousands going down here. In other words, when we take a lot of carbohydrates from our diet, some of the carbohydrates are used to form glycogen, and we can store it in our cells. especially our skeletal muscles. And what happens then? When you are exercising, when you're having a game or a meet, the skeletal muscles already have this storage of glucose. So then when it needs energy, it just breaks up a glucose molecule inside the cell, put a little oxygen in it, and now I can undergo the Krebs cycle and get a lot of ATP or energy. Perfect, right? The problem with this is that there's a limit to how much you can store. It's like a gas tank, right? What happens if gas right now goes to 99 cents? Well, everybody will try to get as much gas as they can, but there's only so much you can put in your gas tank. So no matter what, if we eat a lot of carbs, we can only produce so much glycogen because you only have so much skeletal muscles. If you eat more, and now you have excess amount of glucose, and now your stores of glycogen are completely full. Your gas tank is full. What are you going to do with the rest? Right? Well, if it's 99 cents, you might buy another gas canister, right? An external one. In our body, we don't have another gas canister. But what we can do is take any excess glucose and turn it into fat. As you turn it into fat, there's no limit to how much storage of fat we can have. Keep in mind, if you've ever watched My 600-Pound Life, right, you will see how big people can get. And there's virtually no limit to how big those people get. Some of them are up to 1,000 pounds, right? And it's not from glycogen. It's from overeating. Then they fill up your stores of glycogen, and the excess sugar beyond that will be converted to fat. And then you're going to transport it to adipose cells, and now you're going to add more fat to your fat cells. So look at these carbohydrates, and we'll talk about these. You have three main monosaccharide or simple sugars: fructose, glucose, galactose. If you look at glucose and galactose, they look almost identical, right? So each point is supposed to be a carbon. All three have the same chemical formula, C6H12O6. The difference between the three are the location of the bonds. The only difference between glucose and galactose, glucose right here, oops, right, glucose right there, right? The only difference is carbon number 4, the hydrogen sticks to the right, carbon number 4 here, the oxygen, the hydroxyl group sticks to the left. So that's the only difference. Now the weird thing is this: we have glucose receptors, we have galactose receptors, we have fructose receptors. So when we eat fructose, where do we get it from? When you eat like a grape and it's very sweet, it has a lot of sucrose in it. Sucrose is a combination of this one and this one, glucose and, so it's a combination of glucose and fructose. That's sucrose. Most of what we see in nature are disaccharides, right? When you drink milk and you get lactose intolerance, right? Where's that lactose from? Lactose. Let me see if I have a picture of lactose. Right, so here's sucrose, the table sugar, the one that we see in plants as well. What do we see? Right, it's made from glucose and fructose. You have a dehydration reaction, and you build something bigger. The weird thing is this: when it's connected to each other, you can't absorb it. You may remember, I just said we have a glucose receptor, we have a fructose receptor. Why can't we absorb it if we have the receptors? The thing is, our receptors are so specific that when the glucose and the fructose are bonded together, it can't bind to the receptor, any one of them. So in order to absorb that sucrose, we break this reaction apart, break sucrose apart, and have the reaction go backwards. We can absorb each one independently. So what I want you to know is just the types of sugars that we see: sucrose, glucose and fructose. Lactose, glucose and galactose. Now, this lactose one is an important one, especially in certain cultures, in certain population groups. For Asians, this is very important, this lactose. There's a lot of lactose intolerance. That lactose intolerance is different than a milk allergy. Lactose intolerance means you're missing the enzyme, lactase. Lactase breaks apart the bond between glucose and galactose. As it breaks it apart, I can absorb glucose, I can absorb galactose because I have receptors for each one. But when they're combined, nothing gets absorbed. It stays in your GI tract as lactose. And now you have a solute in your GI tract, lactose, that usually gets absorbed and now isn't. What happens is water is drawn towards it. As water keeps and stays in the lumen of the GI tract, now you have water in your GI tract. What do we get? Diarrhea, right? Now you have glucose and sugars as well, the lactose, also found in the lumen of the GI tract along with the water. The lactose becomes great energy for the microbes in our gut. So they produce methane in response. And now we have methane as well as diarrhea. You have that explosive diarrhea that some people get with lactose intolerance. Now, what do we do for those people? Give them a lactose pill. Lactose pill is man-made enzyme for lactase. So what it does is it replaces our own enzymes meant to break this lactose molecule apart. All right, so here's starch. Glycogen is the animal form of how we store starches, right? Now keep in mind, starch right here is nothing more than glucose molecules bonded together. The weird thing is this, you see how this glucose molecule has an oxygen bond right here? We call this, because it goes downwards like this, an alpha glycosidic bond. We have enzymes that can break this apart. Now, some of you guys have heard of cellulose. Cellulose is fiber, right? Let me ask you a question. When you eat steak and potatoes, the next day when you go number 2, or late at night when you go number 2, when you poop it out, does it look like steak and potatoes? I know it's gross, right? No, it doesn't, right? It doesn't look like anything. Why? Is because we're able to digest it, break it apart, and then absorb a lot of it. What happens when you eat corn? What happens when you eat a salad, right? What happens is this. Salad, corn has, instead of starch, has a lot of cellulose. And because we can't break apart cellulose, it comes out looking very similar to what it came in looking like. It's because we don't have the enzymes to break it apart. The weird thing is, even though cellulose comes out almost whole, it's made from the same thing that starch and glycogen are. It's made from glucose molecules bonded together. The only difference is that the link between the glucose molecules doesn't face down, but faces upwards. All right, so instead of facing down, it faces upwards a little bit. That's enough, right? So then our enzyme can't get in there and break it apart. And because our enzymes for starches can't break it apart, then it comes out looking very similar to what it came in looking like. I know, kind of gross. All right, so fats, most of the time when we think of fats, we think of this, right? The fats that we store in our fat cells when we gain weight, that fat is triglycerides. Triglycerides are a combination of a backbone, 3-carbon sugar called glycerol, and with each carbon, a fatty acid attached to it. That's 1, 2, 3 fatty acids and glycerol. By the way, you guys have heard that when we burn fat, we produce twice as many calories. We have more energy produced when we burn fat. Why? It's because when we burn fat, this fatty acid can be used to drive the citric acid cycle. This can also be used for energy. This can be used for energy. This can be used for energy. We're going to see that all four constituents of triglycerides can be used for energy production. In carbohydrates, right, you can only run the Krebs cycle twice. For triglycerides, you can run it 1, 2, 3, 4 times. Twice as much, right, ATPs will be formed. That's one of the reasons why our body, there's almost no limit to how much we can store fats as triglycerides. Because we can store so much of it, and when you burn it, you can actually produce more energy. Now, this is true. Our body loves doing that, especially, and our body hasn't evolved as much as our diet, right? If you imagine, I want you to think of our body and what I just mentioned, but what would happen if instead of living in 2020, we live in 1220 or 1020, meaning 1,000 years ago, 500 years ago even. What happened is food wasn't as readily available. And when you want to eat and you're really starving, you have a tendency to overeat. And then you might not eat for a couple more days. So whatever you eat, you have a tendency to overeat. When you overeat, you produce a lot of fats. The thing is, because you're not going to eat in a couple days, you might burn that fat that you had accrued. And then by the time you eat again, whatever fat you had available from the 3-4 days before, you had a good meal. Well, that fat is now burnt away. And now you feel hungry, famished, and now you're going to overeat again when food is available. Right? That's how our bodies works. Right? That's how the body wants to work. The problem is now our body still wants to work in that kind of direction. Overeat when there's food, so then you can store for when there isn't food. Nowadays, there's always a little bit of food. So we overeat when food tastes good, and there's never a time where we're fasting for 3-4 days at a time. So you never burn out all that fat that you had just made. What do you do then? You eat the next meal, and now you accrue even more fat and even more fat, right? And you can see that, especially in people like in the My 600 Pound Life people. It becomes a bad feedback system, right? Now, besides fat, most of you guys have heard of saturated and unsaturated. Saturated fat has a really, really bad kind of label on it, right? Nobody wants saturated fat. What do you mean by saturation? Next time people ask you about saturated fat or tell you about saturated fats, have them really explain it. What is the difference between saturated and unsaturated fat? Most of the time they'll tell you that, well, one's plant, one is animal fat. Yeah, but that's just that doesn't explain the name saturated and unsaturated. You're just telling me the location of the fat, right? The difference between saturated fat and unsaturated fat is this, and it gets really scientific. The thing is, saturated fats have all these carbons, and it's saturated with hydrogens. See that? All these hydrogens. Every single carbon has the most amount of hydrogens that can be bonded to it. In an unsaturated fat, you might have a double bond here and here and here, meaning that you don't have hydrogen, the maximum amount sticking out. That's the only difference between saturated and unsaturated. Now, studies have shown that if you eat like pure lard, that's not good for you, right? But if you do eat fats, animal fats, every now and then, it's not as harmful as you would imagine, right? Now, there have been studies, and there was one big study in the 1970s that really made this fat, all unsaturated fat, almost give it like a black label, a black eye. And the thing is, that study basically has been kind of debunked because the person making the study and his group actually pinpointed certain population groups. So they saw people in Asia that ate lots of fats, right? Lots of animal fats. They saw that they eat fats, but yet the lifespan was very high. They weren't included in the actual study, right? But when they saw populations like in Mexico, where there's a lot of fats eaten and the population of the elderly and the death of elderly, right? The age of death was lower, they added that to the study. So they pinpointed their actual data, which is crazy. And at UWM, we kind of had to, we wanted to take that information and really kind of learn from it, right? Because for the last 60 years, we've been kind of going through this craze about being super scared of just animal fat, especially in America, right? Now, where else do we see lipids? As part of our lipid bilayer in the form of phospholipids. And then fats can be in the form of steroids, sorry, right here, right? So steroids, take a look right here, cholesterol. You guys know we take our cholesterol levels. About half of the cholesterols we have in our body are made by our body. The reason why is simple. Cholesterol has a couple of different important functions. Take a look what cholesterol is there to make. Estrogen, testosterone. See that? Everything, these two incredibly important hormones are produced from cholesterol. Now, if you look at estrogen and testosterone, if you look at this right here between estrogen, it's almost identical, isn't it? Right? This and this look very similar as well. Why am I mentioning it? You have heard of people taking anabolic steroids, and it vastly increases their testosterone level. Why do we see some of them start to develop breasts? Because when your testosterone levels are that high, some of it gets converted to estrogen, right? By an enzyme called aromatase, this can get converted to that. Kind of crazy, right? And the same thing with women, all right? Women that have high estrogen levels can have some of that estrogen converted to testosterone as well. So high levels of both are not good. You can be converted one to the other. Now, bodybuilders are crazy. So what they'll do when they're taking anabolic steroids is that they'll actually take another medicine that blocks that aromatase. So then they don't have this estrogen production from the overproduction of testosterone. Right? But cholesterol is required to produce our main sexual hormones for each males and females. Now, go into more detail when we start talking about, right, with my videos. I'm going through this kind of fast because we don't have as much time as we do in a normal semester. What I want to talk about last, and then give you guys a little break, is proteins and protein structures. Now, with proteins are made out of amino acids. Amino acids are then bound, one amino acid next to another one, bound together in a bond we call a peptide bond right here. Here's a peptide bond. Then we're going to add even more amino acids to this. So then you have another amino acid here, and then another one over here, and then another one. Amino acids can be proteins can be thousands of amino acids in length, right? So what we're going to need to know is we need to understand, especially now, and then in chapter 3, understand number 1, amino acids are bonded together to form larger and larger proteins, right? Now, there's 20 different amino acids. You don't need to know the structure. All amino acids have this base area right here that's identical, right? This backbone, carbon, oxygen. This is our carboxylic acid. You have a nitrogen group on the other end, right? This is called an amine group. Amino acid. The difference between one amino acid and another is this secondary carbon right here, right? If it has a hydrogen attached to it, it's glycine. If it has some other larger structure, like this one, is tyrosine, right? So the only difference between one amino acid to another is a side chain that we call an R group. And there's 20 different amino acids. The location of which of the 20 goes in which area will tell us the protein structure, right? So in order to make a protein, you need to know how to make it. How do we find out how to make it? Well, all proteins are coded for in our DNA. You guys have heard of a gene before, right? Most of you have heard of a gene, a DNA gene, and really don't know what it means. All a gene is a coding for how to make a protein. That's all it is, right? So the genes will tell us how to make the different proteins in our body. We have hundreds of thousands of different proteins in our body. Insulin is a small protein. Your thyroid hormone is a small protein. Your muscles, your actin and myosin, when you're contracting a muscle, that's a protein, different proteins. So we have hundreds of thousands of different proteins in our body made from the 20 amino acids. How can we make so many different proteins? Is by placing certain amino acids at certain locations, right? By placing certain amino acids at certain locations, we get a sequence of which amino acids go where, and it starts leading us a path of how to make the protein. That's what we see here. For a protein to have the right shape and function, the first thing we need is what we call the primary structure. The primary structure is the amino acid sequence. Which amino acid goes into number 1? Which amino acid goes into number 2, number 3, number 4, number 5, all the way down the line. Kind of makes sense, right? If you're making and you're cooking at home, you need to know what to make first and how to make it. You need to have your ingredients on how to, all right, and ready to go. So you need instructions. Same thing here. To make the protein, you need instructions. That's a DNA. That's a gene. And then, right, we need to put everything together. We need to find out, all right, when we're cooking something, do we salt it first? Do we marinate it, right? Do we do all these different things to it before we cook it? So we need to know the sequence. And the primary structure of a protein tells us the sequence, right? If the primary structure is correct, usually the other structures take care of itself. So primary structure is a sequence. Which amino acid goes where to make the protein I need? Obviously, if your amino acids are incorrect, you're not going to make the right protein. Now, if the amino acids are correct, we're going to see even a correct protein needs to have a proper shape. Proteins always has a function. It always has a binding site to something. But in order to have that binding site, you need to shape that protein, right? You need that protein to have a certain shape and structure. And the primary structure tells us the sequence. As we have the sequence, then these amino acids start to hydrogen bond to each other. As they start to hydrogen bond next to each other, they form secondary structures. And there's 2 main types of secondary structures. In other words, right, this amino acid chain isn't just one long line. That's it, right? No, as they start forming, they start reacting with each other. And certain areas, right, they start to react and hydrogen bond and form an alpha helical structure, like a ribbon. Not double helix like in DNA, but just an alpha helix single helical structure. In other areas, these try to hydrogen bond and they form a beta helix, right? Or beta pleated sheet structure. Sorry about that. This beta pleated sheet looks like an accordion. And what happens is that secondary structure starts connecting different amino acids pretty far apart. Take a look at how far apart in sequence this amino acid is to this one. There's there's about 10, 15 amino acids in between them. Normally, they're not going to interact with each other. But because of the way they're folded up right here, they start interacting and they start forming a secondary structure. So hydrogen bonds, important for a secondary structure. And these structures will be incredibly important to form the proper folding of a protein. We can have the right amino acids. So our primary structure can be good. But if our secondary and our last one, tertiary structure, is incorrect, then the actual protein is non-functional. In order for the protein to be functional, it needs to have the proper shape and binding site. In order for that to happen, you need to bend it. That's what this is, the bending, right? Now, the tertiary structure is the massive folds that we're gonna see. Now that we put everything together, we're gonna fold this protein so then it has a correct shape for its binding site, right? Now, in terms of protein structure, there's a lot of research going into how we get these folds. And for the most part, we know the proteins that actually helps in these folding. The problem is we still don't really understand how it gets its proper shape almost all the time, right? It's incredible that it happens intracellularly in the trillions of microscopic cells in our body. And the folds occur, and as long as the amino acid sequence is correct, as long as our ribosomes and all the other support proteins are correct, usually the protein you make folds by itself, right? And have this tertiary folding by itself, right? Now, what do I want you to know about tertiary folding then for the test? This is the big fold, where you're taking, let's say you're making a paper airplane. When you're making a paper airplane, you can't just fold your piece of paper in half and expect it to be an airplane, right? You need to make little creases, little small folds first. And then once you make all the little small folds in a paper airplane, then you fold the sides together to form a wing. That's what we see with the secondary folds or secondary structure. Secondary structure are those small folds that you have to put into your paper airplane that then allows us to fold up the piece of paper to make a complete airplane, right? That's a tertiary structure. You're folding up the whole structure, but that can only be folded up correctly if the localized structures or folds are correct. Once you have the whole secondary and localized folds correct, then you can push everything together and form a functional whole protein, right? That's what tertiary is. You're folding the whole protein to have the proper shape and binding site. Now, in some proteins, you have a quaternary structure as well. And the quaternary structure just tells us that you have other amino acid chains working together to stabilize each other, right? We're going to see this especially with hemoglobin, our oxygen binding protein. It's actually 4 different proteins that binds together and forms a much larger, more stable individual hemoglobin molecule. Right. Now, folding is so important that when we unfold the protein, it denatures. Now, what is denaturing of a protein? And when do we see this? When we're cooking an egg. Keep in mind, when you're cooking an egg, it's the egg whites we're looking for. What happens to the egg whites when you're cooking an egg? It turns very, very white and fluffy. Why? Because there's a lot of protein in the egg white, and by producing and by putting heat onto the egg white, all the proteins in the egg white unfolds. As it unfolds, what happens? It unfolds, and as it unfolds, it gets springy, turns different color, right? and looks completely different than what it did before you added heat to it. All right. Now, just the rest of this is just on the protein structure. So review it. And what I'm going to talk about next is just DNA, right? So here's DNA and nucleic acids. There's two main types of nucleic acids. There's DNA and RNA. We'll talk a lot more about DNA and RNA in chapter 3 towards the end, right? And what we're going to see is with the DNA, you have nucleotide. All right, DNA is a combination of a sugar, deoxyribose, a base that sticks out. The base can be thymine, adenine, guanine, cytosine. It can be any one of the four. And then with any molecule DNA, you have a phosphate group right here. This phosphate group forms the main structure of our sugar phosphate backbone of our DNA double helix. So here's DNA double helix right here. The main structure of it is formed from the sugar phosphate, sugar phosphate, sugar phosphate. It forms the backbone of our DNA double helical structure. How about another side? Sugar phosphate, sugar phosphate. See that? The sugar phosphate backbone is what allows the double helix to stay in place. Now, the bases stick towards each other, face towards each other, with adenine always found with thymine, guanine with cytosine. See that? We call that the law of complementary base pairing. This is also important because if we know just one side of the DNA molecule, because if we didn't have this side, if I know one side has thymine, the other base should have to be adenine. If I know over here there's guanine, then the other base on this other side has to be cytosine. Seem all right in reverse. If you know one side's adenine, other side's thymine. By knowing one side, you know the other side. Perfect. Now, they're always found, there's only 4 bases. They add up to 100% because there's only 4. And what we're going to see is because of this, what we call the law of complementary base pair, right here, right? Because of the law of complementary base pairing, where adenine's always with thymine and guanine's always with cytosine. If we know the amount of one base in a region of DNA, we know the amount of all the bases. Here's an example. If in a certain region of DNA, there's 15% adenine in that region, how much guanine should we see? So right here, where's that picture again? Right, meaning right here, right? Let's say this area right here, we see has 15% adenine. Now I want you to know how much guanine is in it. Well, what happens? If you have 15% adenine, that means you have 15% adenine and thymine are found in equal numbers. If I have 15% adenine, 15% thymine. Combined, that's 30%. The other 2 remaining bases have to add up to the 70%. And they're found in equal numbers. So 70 divided by 2 is 35. So you have 35% guanine, 35% cytosine. In other words, by telling you the number for one, you should be able to find out all 4. Just from this law of complementary base pair. All right? Now, any questions? Good. You know what I'm going to do, all right? Actually, I'm not going to take a break now. What I will do right now is just continue for the next 15, 20 minutes, right? And let me see. And then hopefully we can get through some of it and then leave a little bit. Do you guys have any questions so far? That's not it. accidentally logged off my VMware, that's why. All right. What I'm going to do is go through chapter 3 a little bit. I want us to talk about certain structures and certain things. If we can get through DNA, if we can get through, if we can try to get into and through osmosis and filtration, right, we'll be in pretty good shape. Now, especially with chapter 3 material here, right, up your quiz, which I will release this weekend, right, will be, and you will have an assignment on Connect as well, right? Your quiz will be on parts of 2 and from chapter 3 all the way till protein synthesis and DNA replication, right? So just keep that in mind, that it is until like 3.10. So all this about organelles will be found in your quiz as well. So there's, you know, a lot of this should be a little bit of review from just biology as well. So it shouldn't be too new to you. So let's take a look at a couple concepts here, right? The big concept I want us to get into, skip this for a second, is osmosis, filtration, and diffusion. Where are you guys? So right here. So 3.6. I want you guys to understand this concept. What is diffusion? So in diffusion, we have movement of solutes from an area of higher concentration to an area of lower solute concentration. So take a look right here. We have a beaker of water, right? A beaker of water, and we put a salt crystal in there, a salt cube. Give you a sugar cube, it doesn't matter, right? Obviously, in that salt crystal is going to be very concentrated in salt. So when you put it in water, the salt starts to dissolve. As it starts to dissolve, the salt starts moving outwards to an area where there's less of the salt and more of the water. So what happens then, right? All that salt starts to disperse outwards throughout the amount of water in our volume right here. Now, keep in mind with this, right? Eventually, we hit an equilibrium where all the salt right here has completely covered the inside beaker. So then if I take amount of salt concentration in here, it should be the same as a concentration here, as a concentration of salt here and over here, right? Why? Because once you hit equilibrium, there's no more net movement of that solute. Now, why does the salt like to move back and forth? What happens is microscopically, the salt actually is moving around a little bit. As it's moving around, because there's so many other salt crystals here, as it shakes and moves, it bumps up. As it gets bumped to area where there's less salt, way out here. From high salt to area of lower salt concentration. Now, eventually, right, it gets bumped, and then when it's so kind of dispersed everywhere, the salt has no other place to move. Now we're at equilibrium. Now with diffusion is always movement of any solute from high concentrations to low concentration. Now, what can increase the rate at which it moves? The amount and the density of the concentration gradient. Meaning that what would happen if I put this salt crystal, now I put it into this, a beaker already full of salt. What happened is you're not going to have as much salt being dissolved, right? The new crystal, well, that new crystal, there's so much salt in that solution already. So if I put a crystal in, it's usually not going to dissolve much at all, right? Because there's such high amount of salt already in the solution. So concentration gradient is a concentration difference. If it's very similar, there's not going to be a net movement of the solute. Again, diffusion, movement of solutes from high solute concentrations to low. Here's an example in our body. In our body, very few things are what we call, right, are able to pass through our plasma membrane. Things like fats, gases can go right through our plasma membrane of our cell. The plasma membrane of our cell is the border of our cell, made out of phospholipids. Now, lipids and fats like to react with other fats. So when you have a gas, gases go right through. And when you have fats like steroids, they can go right through your cell. That's why anabolic steroids are so effective in gaining weight and muscle. What else? That's usually about it. Fats and steroids are the only ones that can go right through your border, your plasma membrane, from high concentration to low concentration. Everything else need a little bit of help, right? Sodium, potassium, salts, right? Ions need help. Glucose needs help to get in and out. Now take a look right here, right? We have a lot of sodium in pink on the outside of the cell. Very little sodium on the inside. Notice sodium can't just get through, not like the fat molecules here. It needs a membrane channel, a protein found on the plasma membrane. What happens? When the membrane channels open for sodium, sodium follows its concentration gradient. From high concentrations outside the cell, sodium wants to go into the cell. It only goes in when the membrane channels are open, right? When the membrane channel is open, it allows sodium to go through. We call this facilitated diffusion. Why? Because we need to help or facilitate. So then the ion can move through. Lipids can go right through from high concentration to low. We call that simple diffusion. Anything that needs help to get through the plasma membrane requires facilitated diffusion. An ion channel allows us to do this. Here's an ion channel for sodium. When it opens, do we have to put energy in? No. It just goes from high concentration down to the lower area. Perfect. Right? Glucose, it gets brought in that way too. High concentration of glucose, it then has to bind to a glucose receptor, and it goes from high concentration to low concentration, following the laws of diffusion. The law of diffusion is found in chemistry, physiology, in physics, right? It's a natural law. And because it's a natural law, when ions move down the gradient, you always, right, it occurs naturally and without any energy required. Osmosis is what happens when you have some kind of membrane in the way. Now, obviously, our body has membranes, right? Plasma membrane. We also have water channels in our cells called aquaporins, right? So because of the presence of the aquaporins in the plasma membrane, water can move back and forth pretty readily. So here you have an aquaporin, and water should be able to move in and out pretty easily, right? Now, in osmosis, we see osmosis when there is an imbalance of solutes in any one area, right? So let's take a look at what it means here. Let me go back. And I do want you guys to watch my video with me in lecture, because I have a better example of it. I don't like the example of the beaker, and you'll see why in a second. When you have just one beaker, right? So what happens is this, right? I have water, and I'm gonna split it in half. And right here, well, they put okay, now I get what they're doing here. It's kind of weird, right? But they have this kind of, what looks like a plunger that has a little bit of a membrane here, and it's got 3% salt solution. The membrane stops the salt from going into the pure water. What happens then? When I push this, what looks like a little plunger filled with salt water in there, I put it into a large distilled water filled beaker. What happens then? What happens is the salt, because of that membrane, can't leak out. Because the salt can't get through that selectively permeable membrane, you stop that movement, and what happens, right? You stop the fusion. If it wasn't for this membrane, and I just had the plunger and that's 3% solution salt water in there, what would happen is that the salt would just disperse out from high concentration to low concentration. Because of the membrane, the fusion is stopped, right? Water, though, can get in because it's selectively permeable. What happens? Water, from the distilled water with no salt in it, low sodium concentration. Why? Because it's pure water, right? What happens when we put the plunger in? Water can go through the actual membrane and go from low solute, pure water, to a region of high solute, 3% salt solution. What happens then? The 3% salt solution, take a look at how much water it has now than before, right? Eventually, so much water goes in that you fill up that solution, fill up that plunger. That is when osmosis will stop. Right? So osmosis is movement of water from an area of low solute concentration to an area of higher solute concentration. Now the book will talk about movement of water from area of less water to an area of more water, right? Understand it in terms of the solute. That's how I'll ask you questions. Now, let's see what happens right here. How does it affect our cells then? It can affect our cells by drinking too much water. And this is the reason why we don't give you just pure water to drink, even when you're severely dehydrated. If I give you pure water, lots of pure water, you're drinking a solution that is hypotonic. It's got no salt in it. What happens then? Because it's got no salt and no solutes, that water wants to go into areas where there's higher solute concentration. So at first, you drink water, it goes into the GI tract, you absorb it, it goes into your bloodstream. As it goes in your bloodstream, that pure water starts to go into the cells, your red blood cells. Why? The red blood cells, inside of it, has a lot of solutes, 300 milliosmoles. The water in plasma, right here, there's less solutes now. So the water in the plasma starts going into the red blood cells, causing your red blood cells to then blow up in size and then rupture, right? That's what happens when you drink pure water. So I want you to look up, right, winning the Nintendo Wii by holding the Wii. What they did was they had them drink gallons of water and then hold it for as long as they could. Because water, how can it hurt anybody, right? It's water. It's good for you. Well, by holding it, what happens is, right, it allowed that person to decrease the osmolarity of their blood. And then water leaked out into the interstitial fluid, right? And then when that water leaked out into the interstitial fluid, she started having edema throughout her body, fluid buildup. There's only one place where edema is dreadful and life-threatening, and it's just in the brain. And that's what happened to that woman that won, but she died, right? She had water movement into the interstitial fluid in the brain, caused the brain to hemorrhage out of the brainstem through the foramen magnum, and she died as a result, right? That's why the bad thing is this: all she had to do was just urinate. If you let yourself pee, you're able to get rid of any amount of excess water. It's not comfortable because you'd be peeing every 2 minutes, but you're able to get rid of it, right? But she didn't let herself pee because she wanted the Nintendo Wii so bad. We give people isotonic solution when we give them saline. So then we're giving them solution that has 300 milliosmoles of salts What happens then? It's got the same osmolarity as the cells, so there's no net movement of water. Now, what happens if you drink seawater? If you drink seawater, you're taking a lot of salt into your system. You're making your plasma more salty. Because the plasma becomes more salty, because that's where you absorb it from the GI tract, goes into the bloodstream. All that salt is added to your plasma. As it's added to the plasma, now the red blood cells stay at 300 milliosmoles, but the surrounding plasma goes to 400 milliosmoles of solute, higher solute concentration. Now there's higher solute concentration in the water, the plasma, than in the cells. Water always goes from low concentration inside the cell to higher concentration. So water leaves the cells and the cell starts to shrink and die as a response. So the best thing to do, drink pure water, but let yourself pee. Never drink seawater. That's never good. All right. Any questions so far? Let me see if I can find filtration. So I'm trying to find one specific one on filtration. I know I have that on my lecture ones up. Guys, I have to cut it off a little bit. My throat's kind of closing up on me today. I don't know if I'm getting sick or what, if there's a weather change. Do you guys have any questions about this weekend? All right, I will have a review. I will have a connect assignment for you guys to do. It'll be on a little bit of, like most of it's going to be on chapter 3, but a little bit of 2, right? A little bit of 2, a little bit of mostly on chapter 3, right? And then a quiz on chapter 2 and part of 3. Review your organelles. I have videos on that, so make sure you go watch those. And I just want to make sure that you are watching the videos, right? This kind of, when we're going through it together right here, should be just a additional kind of going through material. There's no way we can replace the normally 6 hours of class we have with what we have for an hour and a half or 2 hours, right? So make sure you give yourself time to review the notes as, sorry. Sorry, my throat's closing up. So make sure you're giving yourself time to review new notes, watch the videos. All right. I will send an email announcements about which ones I want you guys to really focus in on. All right. I'll let you guys go. I have to go. So text me, email me if you need anything. All right.